Hello, today we are going to learn about meiosis. So, what is meiosis? Sounds very similar to mitosis, but it's a little bit different. So, it is a type of cell division that results in four daughter cells, each with half the number of chromosomes of the parent cell, as in the production of gametes and plant spores. So, with meiosis, the cells are getting their genetic information reduced in half, so that when it pairs up with another sex cell that also has another half, they can make one whole again, which is what happens in fertilization. You can see in the GIF above. So like sperm cells would have half the genetic info from the male, while the egg cell would have half the genetic info of the female. They come together to make a new person um, that has the full genetic info. Now, there are actually 8 different phases of meiosis. It's prophase 1, metaphase 1, anaphase 1, telophase 1, and then all those happen again as prophase 2, metaphase 2, anaphase 2, and telophase 2. So we learned essentially what happens in prophase, metaphase, anaphase, and telophase in the mitosis video, so if you want some more specifics, I would refer back to that. But first, let me give you a little more information. So, interphase will occur prior to meiosis, but it will not begin again for the new daughter cells after meiosis. Meiosis is like mitosis twice, so not exactly like mitosis twice, but it's similar. Chromosomes will arrange in homologous pairings for meiosis. Half of DNA in sexually reproducing species comes from the mother, while the other half comes from the father. Okay. So, looking at the images above, the difference between the homologous pairings in mitosis and meiosis is seen there. So with mitosis, it's just the replicated DNA all going completely down the equator of the cell, while in meiosis, homologous chromosomes, so basically one chromosome you get from the mom could go on the left side of the equator, while one chromosome from your dad could go on the right side of the equator of the cell. So that's how they initially pair in meiosis. So it's a little difference, but it is a very key and important difference. Okay, so starting meiosis off with prophase 1. Exactly the same thing happens as prophase 1 in meiosis or in mitosis. That nuclear membrane is going to fade away, and then the DNA is essentially going to be coiled tightly into those chromosome shapes. In meiosis, though, in prophase 1, there's an extra process that happens, which is called crossing over. Crossing over is the exchange of genes between homologous chromosomes, resulting in a mixture of parental characteristics in your offspring. So again, half your genetic information comes from your father, the other half from your mother. So what happens is these homologous pairings of chromosomes, they literally swap a little piece of their genetic info with one another. You can see that in the GIF in the top left and in the GIF in the bottom right. So essentially what you're going to have after this crossing over process is that each chromosome that originally came from father is going to have a little bit of the mother's DNA on it, and then all the mother's chromosomes are going to have a little information from the father's DNA on them. Okay, next is metaphase 1. Okay, also very, very similar to the metaphase we know about in mitosis, but there's an additional process. This process is known as independent assortment. So yes, chromosomes are being lined up at the equator of the cell, but independent assortment is when there's a formation of random combinations of chromosomes in meiosis and of genes on different pairs of homologous chromosomes by the passage, according to the laws of probability, of one of each diploid pair of homologous chromosomes into each gamete independently of each other pair. Gamete is just another name for sex cell. We're going to talk about diploid and haploid, those terms, in a future video. But essentially what this means is that when the chromosomes line up at this equator, basically the side that the chromosomes you got from your mother and the chromosomes you got from your father, the side that they are on of that equator of the cell is random, what side they're on. So for example, you randomly could have all the chromosomes from your mom on the left side, and all the chromosomes from your dad on the right side, or they could be flip-flopped in between. So looking at this picture at the bottom, in just the meta 1 line, you can see that if blue is the father's DNA and the red is the mother's DNA, in that first possibility, you have the dad's chromosome on the left, the first one, but then for the next homologous pairings, he's got 2 on the right side. Next possibility is you have 2 chromosomes from the dad on the left and 1 on the right. Then for the next one over, you could have the first one and the third chromosome be on the right for the dad, but then the middle one's on the left. And then the last option again could be like you have all the chromosomes from the dad on the right, while the mothers are on the left. So that's what we mean by independent assortment, is it's just randomly lining up your chromosomes. They're still in their pairings, but as far as which side of that equator those pairings end up being split from, that is random, and that is known as independent assortment. All right, so crossing over and independent assortment are really the extra 2 processes that happen in meiosis. Everything else will be pretty straightforward. So after metaphase 1, where those chromosomes again are randomly or independently assorted along the equator of the cell, those spindle fibers in anaphase are going to attach to the centromeres of those chromosomes and start pulling them towards each pole or ends of the cell. Then in telophase 1, new nuclear membranes form around those chromosomes, and they're almost ready to split with cytokinesis, which you can see in the GIF as well happening there. But again, remember, cytokinesis is technically not a part of mitosis or meiosis. Mitosis and meiosis are all about the nucleus of splitting and dividing. Okay, then we get straight into prophase 2. So once those nuclear membranes were made in telophase 1, they break down again. Prophase 2 now happens. So again, chromosomes are paired up, and they get ready for metaphase 2, which is where again they are lined up, and this is where it starts to look a lot like mitosis now. Then anaphase 2 is going to start pulling those apart. And then telophase 2 is nuclear membranes form around each of those again. So again, with meiosis, you end up getting 4 cells with half the genetic information, and they're all uniquely different from one another. And in mitosis, you get 2 cells with the full amount of genetic information that are identical. So I'm going to make a whole nother video on comparing and contrasting asexual reproduction and sexual reproduction, so be on the lookout for that. Here's essentially the process of meiosis. As soon as the nucleus has been formed, again, just a reminder, so once that nucleus reforms in telophase 2, that's when meiosis officially ends. So this last slide of this video is just for you to see the whole process all at once. There's some good information underneath each of those phases as well. Thank you for watching. Please share and subscribe, and remember to spread friends.